Alright, so let's see how long it takes us to get through. Alright, so today we're going to be talking about RNA, so we're going to be talking about the conversion of DNA to mRNA to eventually make protein. And we're also going to talk about DNA replication as well as mutations today. So if we start on the first page, the central dogma, we know that it says that DNA gets transcribed into mRNA, and then mRNA gets translated into protein. That's not exactly the complete story. So what we're going to say here is that the complete picture is DNA gets transcribed first into hRNA. We're going to say hRNA, well, hnRNA, is heterogeneous nuclear RNA. So this is the first transcript that you get. So this is the initial RNA you create when you transcribe or transcript. Just change it to transcription. And then what happens next is you do RNA processing. And from there, then you get mRNA. And then that mRNA is then translated into protein. So this is the complete picture. So that word there is protein. That cut off a little bit. This is the complete picture of what's going on here. And then we investigated through research retroviruses. Retroviruses are a way of reversing the central dogma. So what do retroviruses do? They find a way of changing mRNA into DNA. So they use reverse transcriptase, so an enzyme, which allows them to do this. Remember, the central dogma is not completely just DNA straight to mRNA straight to protein. There's that step in the middle where we make first hnRNA, which is the rough copy of RNA. We process it, we perfect it. We cut out the introns, we put a 5' cap on, we put a poly A tail, and that's what we use as mRNA. And then that's translated later into protein. Okay, so we'll be going over this process today, talking about the different parts of it. All right, so transcription. Now remember, we're going to say in transcription, we read the DNA strand in the 3' to 5' direction, but then we transcribe or transcript in the 5' to 3' direction. Remember, it's all anti-parallel. Okay, then RNA processing in eukaryotes, hnRNA, which is made first. Remember, this is the original copy. It's processed into an mRNA transcript before it leaves the nucleus. And then translation, tRNA bind to the mRNA transcript with the help of ribosomes and polymerize amino acids. So basically, we are adding amino acids together by peptide bonds. So that's what we're talking about here, peptide bonds. And then even after you make the protein, you can do post-translational modifications. So that protein can be modified even more. So here we can add a lot of things to it. So we can do adding of sugars, lipids, or fats to those proteins that we make. And then some of that protein can be packaged by the Golgi apparatus. Then it's packaged into vesicles. And they exit the cell. By exocytosis. So basically, that's a majority of what we're going to be talking about today. So these 4 major ideas. Okay, so if we're looking here, we're going to say that this first rough copy here is your hnRNA. it gets processed, so we're going to have mRNA. And then here what we make is we make our primary structure. So remember, primary structure is just a bunch of amino acids connected by peptide bonds. But that protein does not become active until it reaches its tertiary form, its tertiary structure form. So now let's talk about the difference between eukaryotes and prokaryotes. So in eukaryotes, our mRNA, we're going to say is monocistronic. So it codes for basically one single polypeptide. So we're going to say one mRNA codes for one polypeptide. So one chain of amino acids connected together. So this is our polypeptide chain here. But we can get around this problem by doing RNA splicing. So RNA splicing gives us different combinations. So one mRNA, depending on how I cut it, can give me more than one type of polypeptide chain. I can get maybe 3 or 4 different polypeptides from one mRNA. Now, prokaryotes are different. They're not monocistronic, they're polycistronic. Okay? So from one mRNA, you can make a bunch of types of polypeptides. That's one major difference between prokaryotes and eukaryotes. Another major difference here, prokaryotes, they transcribe and translate genes simultaneously. That's because they don't have a nucleus. What they have instead is a nucleoid region. So there is no RNA processing. So no mRNA processing. So what happens is your RNA polymerase starts to transcribe the DNA to make mRNA, and immediately that mRNA gets threaded through ribosomes. And the ribosomes automatically start to make proteins. So there's no time to process the mRNA being created. As soon as it's made, it goes right into a ribosome and you start to make protein. We, on the other hand, have a nucleus, so we have that middle stage where I told us we have first H and RNA, and then we process it so we can get a better mRNA from the process. Okay? But for prokaryotes, they go straight from this to this to this. They skip that H and RNA stage altogether. So this right here would just represent a prokaryote or prokaryotic environment. And this would be a eukaryotic environment. So that one mRNA strand is being translated, and depending on how they translate, we can make different types of polypeptides. Okay, so transcription. You're going to find that transcription and translations both have the same type of steps. So in transcription, we have initiation, elongation, and termination. So step 1 is initiation. So basically RNA polymerase associates with a template strand of DNA. We're going to say here helicase is the enzyme that cuts the two strands. It makes an incision there. And then we're going to say here topoisomerase relieves the strain on the DNA, so it stops supercoiling. All right, and then here, this allows RNA polymerase to bind the promoter to the promoter region. Now remember, we also have single-stranded binding proteins. These keep the two strands apart. So remember, our different types of nitrogenous bases, we have G to C, and we have A to T. How many hydrogen bonds are between G and C? 3. And then A and T, you only have 2. So we're going to say here, most eukaryotic promoters have a TATA box. That's where we have a lot of these A and Ts. There's only two hydrogen bonds between them, so they're much weaker. So we target that section where it's easier to cut. And transcription factors, proteins that help RNA polymerase bind to the promoter, and eukaryotes. So we're going to see different things today in terms of transcription factors. We're going to say here that we have what's called the sigma factor. So this subunit detects the -10 and -35 elements of promoters and helps RNA polymerase bind in prokaryotes. So the way you think about it is like this. You have a strand of DNA. And we're going to say here that this is the promoter site. And this is the DNA that you want to transcribe. Now think of your RNA polymerase as an airplane, and it's trying to land. Now to be able to land as a plane, you need gears, you need wheels. So when you land on the strip, you don't just destroy the plane. Think of the sigma factor as these wheels that come out of the plane and they target that promoter site. So this plane is landing. The plane is your RNA polymerase. The wheels are the sigma factor. They allow the RNA polymerase to land on the promoter site. Then we're going to say here we have RNA polymerase 1, 2, and 3. So here this allows us to synthesize rRNA, so ribosomal RNA within the nucleolus. Here, RNA polymerase 2 allows us to do hnRNA, so the initial RNA that we make. Also, it helps us to do snRNA. So we didn't talk about this yet. This is your small. So small cytoplasmic nuclear RNA. Okay, so what is this involved in? This is involved in RNA splicing. So remember, RNA splicing, this happens when we're doing RNA processing. Helps us to go from hnRNA to the mature mRNA. And then RNA polymerase 3, it helps us to synthesize tRNA plus a little rRNA. And then remember here, tRNA is just our transfer RNA, which brings in the anti-codon during translation. So if we take a look here, so here we have RNA polymerase, this big thing, and it's coming down, and remember, it comes down on the promoter site. So we're going to assume that this is the promoter site. And remember, the only way it can do that is because of the sigma factor. The sigma factor scans and finds the promoter site so that the RNA polymerase can land there. All these bubbles here, these are just our transcription factors. So they help us find the promoter site easier, so that we can have the RNA polymerase land there and start transcription. Now, step 2 is elongation. So RNA polymerase catalyzes the addition of nucleotides on the growing 3' end of the RNA transcript. So remember, this is our DNA. This is our template strand, which is what we're going to be transcribing. We read it from 3 to 5, so we're going this way. And what we do is we start laying down our mRNA that we're creating, and it's placed down 5' to 3'. Okay, so this will be our mRNA that we're going to make from the template strand. And actually, technically, it's not mRNA at this point, it's hnRNA. It'll be processed into mRNA later. And then finally, the last step is termination. So in that one, we put a poly A tail, and we put a 5', well, we put a poly A tail on that, and then when we're doing processing, we'll put a 5' cap later. So remember, we have the coding strand, which is not transcribed, and then we have the template strand, which is transcribed. So eventually we're going to get this as our mRNA. So we're going to say here, this mRNA, who is it complementary to? To the template. So remember, when we're saying complementary, we mean like they match up with their opposite nitrogenous base. A goes with U. Remember, we don't use T with mRNA. T goes with A, C goes with G, G goes with C. So if these two strands here are complementary to each other, what is the coding strand complementary to? Besides the template strand, it would be complementary to anti-codon. Well, that's later on, okay? That's when we're getting into translation. When we talk about it now. All right, so we've created our hnRNA strand. Now we have to do processing of that strand. Okay, so this whole part is just going from hnRNA to mRNA. Remember, this is the original copy, which we don't want. We got to process it before it leaves the nucleus. Remember, we're talking about eukaryotes when we're talking about this processing. Prokaryotes don't have a nucleus, so they never make hnRNA. They just have mRNA the whole time. All right, so here we're going to put a 5' cap on it, which is made up of a bunch of modified guanines. And then we have a long string of adenines, so that's the poly A tail. Okay, so now we're going to say here the poly A tail is important for 2 reasons. 1, it acts as a lit fuse. So as soon as it leaves the nucleus, it starts to break down. So as soon as it leaves the nucleus, it starts to break down. Why is this important? Because this leads to the second part. It leads to protein diversity. Which leads to cell differentiation. So I'll explain what that means. Did you guys write that down? So we have to think of it like this. You have mRNA that you just created. If it doesn't break down, the mRNA is going to be always hanging around in your cell. And if mRNA is always hanging around in your cell, there's a chance that ribosome will connect to it. Okay? So if a ribosome is connecting to it, it's going to start making protein. And if it never breaks down, it's going to make the same proteins over and over and over again. But this is not a good thing, because you don't want to make the same protein all the time. You want to make different types of proteins because your body requires different types of proteins to do different types of functions. But if you have a bunch of mRNA floating around, you're going to decrease the amount of available ribosomes. You're going to decrease the different types of proteins you make, which means you're no longer going to have specialized cells. All your cells will be the same type. And as a big organism, you don't want that. Because what if something bad happens within your environment? Those proteins that create those cells may all of a sudden not be beneficial, and your whole species is wiped out. Okay, so that's why poly A tail is important, because it allows the mRNAs to break down. They have a shelf life. As soon as they break down, we can make new mRNA to make new proteins, which makes new cells. Okay? So just remember, all of that's connected together. So that's the importance of the poly A tail. It allows for cell differentiation. Questions on what I just said? I said a lot. All right. Okay. So 5' cap put on one end, poly A tail put on the other end. Now we're going to have RNA splicing. So basically, we're going to start chopping out parts of the RNA. To do this, we use a spliceosome. So remember, within H and RNA, you have two things. You have exons and you have introns. So X means they're exiting the nucleus. So the exons are going to stay within the RNA. The introns, they stay inside the nucleus. So introns are cut out, exons are allowed to leave. Okay, so spliceosomes excise or take out your introns. And then what's left behind are your exons, what it helps join together. And to be able to join them together, we use ligase. The enzyme ligase. It helps to glue them back together, the pieces that we cut out. So here, when else do we use ligase? DNA replication. It connects together the Okazaki fragments. Okay, so we're going to see that again later on. All right, so basically before we talk about these portions here, just realize what do we have? We have our pre-mRNA transcript, our H and RNA. It has exons, introns, exons, introns, exons. Remember, exons are going to be allowed to leave the nucleus. Introns have to stay behind. So what does it do? This huge thing here is your spliceosome. So your spliceosome is made up of snRNA, which we said was made by RNA polymerase II. It works with something called SNRPs, or small nuclear ribonucleoproteins. They together make the spliceosome. And what's happening here is this whole structure is those two things together. It's cutting out the introns, because it wants to keep the introns inside the nucleus. But then you have gaps. If you cut those pieces out, now the exon is not connected to this or this. That's when you use the ligase to connect them together. So now this is your new mature mRNA. But remember, eukaryotes are monocistronic, so maybe we don't put all the exons together. Maybe instead we decide to put them in different orders. Maybe we do 1, 2, 3, or 1, 3, 2, or 2, 1, 3. We can do different combinations to make different types of polypeptide chains, each having a different function. All right, so here, I already said this, it acts like a lit fuse. All right, so the start codon AUG always begins the coding sequence. It codes for methionine in eukaryotes and N-formal methionine in prokaryotes. So we talked about amino acids last week. So who could tell me what methionine looks like? Remember, you only have to know the R group. So we know that it's going to have a carboxyl group, an amino group. What's the R group look like?
It has a sulfur.
It has a sulfur in it, okay. Well, what's the rest of it? How many carbons before they hit the sulfur? Okay. And then carbon. That's methionine. That's your R group. Okay, so for eukaryotes, AUG is the start codon. It always codes for methionine. So methionine will always be the first amino acid that's laid down to begin your primary structure. Now, prokaryotes, they don't use methionine, they use N-formal methionine. So let's talk about that. What the hell does that look like? So what does N mean? What do you think N means? So N means nitrogen. It means that a formal group is connected to the nitrogen. So it would look like this. So the R group is still the same. And we have NH now. We don't have two H's because one of the H's is going to be replaced by formal. Formal is another way of saying aldehyde. So that would be N-formal methionine. So formal just means aldehyde. Carbonyl connected to an H. So when it comes to translation, the first amino acid that's brought in for eukaryotes is methionine. For eukaryotes, for prokaryotes, it's N-formal methionine. Okay, so it's just a modified methionine. And again, for eukaryotes, we can make different arrangements of your exons to create different types of polypeptides. So because of this, you might start out with 25,000 codons, but you can end up with 100,000 different types of polypeptides. So this is a common example that's used. Remember, eukaryotes, like we said earlier, are monocistronic. So they use RNA splicing to come up with different arrangements of their codons to create different types of polypeptides. Okay, the stop codons that you need to know. So the start codon is AUG. The stop codons are UAA, UAG, and UGA. They don't code for an amino acid. But instead a release factor that helps to stop the polypeptide chain from forming anymore. Okay, so here if we're looking at a mature mRNA, we have our 5' cap. We have UTR, which is your untranscribed region. Here we have our start region of our mRNA, which the ribosome will read to help make the protein chain. Then our stop codon is here somewhere. Again, more untranscribed region, and then poly A tail. So that's our mature mRNA with eukaryotes. Questions on this before we move on? Keep going. All right, so we have translation now. So translation is just like transcription in the sense that we have 3 steps. Again, it's initiation, elongation, and termination. So remember, tRNA is just your transfer RNA. It brings in the anticodon with the amino acid. So tRNA looks something like this. Now, let's take a look here. If we take a look at this tRNA, here's the 3' end, here's the 5' end. So this is 3' and this is 5'. Here, this is your mRNA that's mature. And remember, when we're doing transcription and translation, we read in what direction? 3 to 5, and we lay down in what direction? 5 to 3. What's the problem here? They're in the same direction. Here, it's reading 3' to 5', but it's also 3' to 5' here. Not allowed. We need to find a way of reversing this. So we're going to say with tRNA, we have actually twisting right here. So what happens, it twists at the end so that this becomes 5' to 3'. Okay, so we're going to say here, doing that, it's kind of like having, we're going to say it's similar to the alpha helix of a protein chain. What kind of structure is an alpha helix? Is it primary, secondary, or tertiary? Secondary. So basically a tRNA can take on a secondary structure. And it has to do this so that we can lay down 5' to 3'. So now, if we take a look here, we're going to say we start out with amino-acetyl tRNA, synthase. It catalyzes the transferring of an amino acid to the tRNA. We're going to say it charges a tRNA. And when I say charges, that means it adds an amino acid to it. So here's your tRNA. We use this enzyme here, and the enzyme allows us to put an amino acid onto it. So once that amino acid is on the tRNA, we say the tRNA is charged. Now the tRNA is going to go to the ribosome to bring that amino acid over. Now, the ribosome here, so a structure of protein and RNA that synthesizes polypeptides from mRNA. So we have 3 sites in terms of this. Bless you. So we have the A site, which is your amino acyl site. This accepts the charged tRNA, so the tRNA comes into the A site. Then we have the P site. So that's the polypeptide site where we hold the growing polypeptide. And then we have the exit site, E. Now, what we need to realize here is that if we're looking at this crazy looking picture, we're going to say that this is your mRNA strand, this thing. And then this big green thing here is the ribosomes. The ribosome is made up of 2 parts. It's made up of the smaller subunit, and then it's made up of the larger subunit.
Do we have to know the numbers?
Yes, you do.
30, 60.
Yep, you need to know all that. So you have the smaller subunit and the larger subunit. So when we're talking about prokaryotes, we're going to say we have 30S, 50S, and 70S. So we kind of talked about this last week. The odd ones are for prokaryotes. So we're going to say here that the 30S is the smaller unit, smaller subunit. The 50 is the larger subunit. And both of them together actually create this larger subunit. And then for eukaryotes, we have what? 40S, 60S, and 80S. So again, this is the smaller, this is the larger, and this is both of them together give me this. You have to memorize them, and you have to remember the order, how they combine together to give you the whole structure. Now, here, the S stands for basically the sedimentation rate. You don't need to know the exact name, it's like a German name, but just realize that it's S stands for sedimentation rate. So basically what happens here is you have let's say you have a cylinder of this stuff, suspended in solution. So these are the different ribosome units in there, the larger one and the smaller one. You put it in a centrifuge, you spin it down. So the S is just measuring how quickly the ribosomes fall to the bottom once you use a centrifuge. Okay, so that's where the S comes from. Questions on this part? So here, so for initiation, all right, so here we're going to say, first thing in initiation, we're going to say the small subunit binds the charged initial tRNA, aided by proteins called initiation factors. So initiation factors help, basically, the smaller subunit to attach to your mRNA. Now here we're gonna say it's different for prokaryotes and eukaryotes. So we're gonna say that the initiation complex that helps with translation, it attaches to the 5' cap in the eukaryotes. Then it scans down the mRNA to find the start codon. But in prokaryotes, we don't have a 5' cap because we never do RNA processing. So instead of having a 5' cap attaching to it, we have the Shine-Delgarno sequence. So this is just a series of amino acids that it scans and reads for, and that's where it attaches. So just remember, the initiation happens at different places depending on if you're a eukaryote versus a prokaryote. All right, in both, once it attaches to these different structures, it's just scanning to see where is my start codon. Once it finds the start codon, then the first tRNA can come in with methionine for eukaryotes or N-formal methionine for prokaryotes. Okay, so then here the large subunit binds to the small subunit and the initiation factors dissociate. So once we find the start codon, the initiation factors disappear, and then these two pieces combine together into a bigger piece, and it slides down and scans and translates the mRNA. So initiation factors are just there to help us find the start codon.